one slipper, one man. Again, in the description there will be any corrections or extra info, specifications, whatever. There was some stuff that I didn't include just for time. I recorded this before I went to work, so yeah, we can stay shaggyful. Peace out. Hello ladies and gentlemen, welcome to your 6th Essential Genetics video on post-transcriptional modification. Do forgive me, there is a toddler having a tantrum in the background, maybe, in this video. Little bastards don't even pay the fucking rent. Fucking little dick. I should have started this out of a song or something, actually. Oh well. This stage only occurs, well, this is a part of protein synthesis. It's a stage that only occurs in eukaryotes, that's single cells, that's cells with a nucleus around them. This doesn't occur in prokaryotes, therefore. Cells without a nucleus, like bacteria or archaea. Last time we covered transcription. Now, after this process in eukaryotes, we go on to this stage. If it's in prokaryotes, the mRNA needs to be transcribed before it can be translated into the protein structure. Something I forgot to mention last video was that transcription and translation happen at the same time in bacteria, in prokaryotes, sorry. So, yeah. Well, let's get into this. We've got our pre-mRNA here. This is the 5' end, this is the 3' end. At this end, something will be added called a 5' cap. 5' cap. And what this is a guanosine, not a guanine, a guanosine. A guanosine is a DNA nucleoside, if you don't know what that is. It's in the first video in this series, I believe. But it's got a methyl group on position 7, I believe. So it's a guanosine with a methyl on carbon 7. So the 5' cap is called 7. I've written this down. Methyl guanosine. And on the 3' end, we've got something called a poly-A tail. And a poly-A tail is just a bunch of adenines. A bunch of adenine nucleotides strung together. To do this, the 3' tip of the mRNA strand needs to be removed and replaced with this tail. Now, what do these actually do? They stop the mRNA strand from being broken down or degraded. The 5' cap stops 5' to 3' exonucleases from breaking up the mRNA strand, and the poly-A tail stops 3' to 5' exonucleases from breaking down the mRNA. The cell has uses these to break down viral DNA, but it can break down your DNA accidentally as well, so it puts these modifications on. If we compare a eukaryote mRNA with a prokaryote mRNA, so this is the prokaryote one, this is the eukaryote one. These white bits here are called exons. These black bits are called introns. Introns are the pieces of RNA that are going to be used to to determine the structure of the protein in translation. In prokaryotes, this is just all the gene. That's it. In eukaryotes, in between these white bits, in between the exons, we've got in between the exons, there's introns. These don't determine the structure of a protein. Instead, in a process called RNA splicing, these are taken out. So, RNA splicing, and that just makes an mRNA strand. That's just pure exons. Yeehaw. The thing that removes the introns is called a spliceosome. So, by spliceosome, don't know if I'm spelling that right. And the spliceosome is made up of what's called small nuclear RNAs and proteins. It makes this a thing that removes the introns. We can also have a process called alternative RNA splicing. Spelled fucking splicing wrong there. So, just a badly drawn RNA. Normally in RNA splicing, all the exons are in one. Exon 1, 2, 3, 4, 2, 3, 4, plus exon number 1, 2, 3, 4. But in alternative RNA splicing, so norm splicing, but in RNA splicing, okay, be patient, you cunts. The introns are removed, but the exons can be split across multiple strands. So we can have exons 1 and 3 split together, and exons 2 and 4 spliced together. These will be translated separately, so you get two proteins from one gene. This does not occur in prokaryotes. I'm actually in a rush for work, so if I can have to get through this pretty quickly. It's not a long video anyway. The spliced, before we had pre-mRNA in eukaryotes. The reason in eukaryotes we call it pre-mRNA instead of just, in eukaryotes. In eukaryotes, before this process we call it pre-mRNA, and then some sort of splicing. Then it becomes mRNA. Also called the mature transcript of mRNA. Again, this could be called the primary transcript of mRNA. Again, in prokaryotes, we just call mRNA mRNA because there's no modification whatsoever. In eukaryotes, before splicing, we call it pre-mRNA. After splicing, we call it mRNA, or the alternative names there. Just to go back to the 5' cap and the poly A tail. These do have other functions, but we don't need to know about them in this video, honestly, you know. All modifications take place in the nucleus of the cell. So to draw you a simplified eukaryotic cell, that's the nucleus. This space outside the nucleus is called the cytoplasm. Nucleus, cytoplasm. And now, the mRNA can move into the cytoplasm to take part in transcription. Anyway, that's us for today. A short video. Nowhere near the length of the fucking essential chemistry 5. That was a disaster. Fucking took up fucking loads of my data. Anyway, I'll see you cunts next time. Peace out.